All right, I think we are going to start. My name is Valuš Dobrák. I'm Director of Molecular Imaging Laboratory at Beckman Institute and running Nuclear Imaging Laboratory. And we don't have too many such talks about the nuclear imaging technology. And today we have a special guest, which is the Yubin Miao. He's an assistant professor in College of Pharmacy in the University of New Mexico. And he will be talking about the novel receptor-targeting radiolabeled peptides for cancer imaging. So we got kind of a theme of with an interest of cancer imaging. And today we'll be talking about the radiolabeled peptides for imaging cancer. And just brief biography, Dr. Miao received his PhD in radiopharmaceutical chemistry from Beijing Normal University. And he did the postdoctoral training at the University of Missouri, Columbia. And right now he's the assistant professor working with the SPECT PET imaging of cancer to detect with the radioactive peptides to detect primary and metastatic tumor lesions non-invasively. So I'm very glad to have him here. And please greet him before the presentation.
Well, thank you so much for your kind introduction. It's my great pleasure to be here and share with you our work on novel receptor targeting, radiolabeled peptides for cancer imaging. So in this talk, I'm going to have 4 sections. In the first section, I'm going to give you a brief introduction on principle of molecular imaging of cancer. Then I'm going to give you 2 sets of examples, but basically, one set example will be targeting the GnRH receptor for breast cancer imaging, then give you 2 generations of the peptides targeting the MC1R for melanoma imaging. So let me start with the principle of molecular imaging of cancer. So as we know, to perform molecular imaging, you need to have 3 components, namely molecular target, imaging probe, and imaging modality here. So both intracellular or cell surface targets can be used as molecular targets. Compared to the intracellular target, there is an advantage associated with the cell surface target. That is, the radiolabeled probe or imaging probe doesn't have to penetrate the cell membrane to reach the target. Once you determine the molecular target, then you need to have an imaging probe specifically bind to the target and generate signals for different types of imaging modalities here. So normally antibody, peptide, or small molecules can be used as imaging probe. Depending upon which imaging modality you're going to use, you need to label this imaging probe either with radionuclide for SPECT PET imaging, or you can label with the optical tags for optical imaging. So in today's talk, I'm going to focus on radionuclide imaging, basically on SPECT and PET. I think I'm going to focus on the SPECT. So by definition, SPECT means single photon emission computed tomography, and PET means positron emission tomography. This slide shows you the peptide receptor binding specifically we used in our projects. So basically you can see the receptors are overexpressed on the cancer cells, and you can use peptide to specifically deliver either diagnostic radionuclide or therapeutic radionuclide to bind to the receptor for cancer imaging and treatment. And as you can see, peptide, the radionuclide can attach to the peptide through a linker. Okay? So different type of linkers can be used to optimize the binding affinity of the molecule, as well as modifying the biodistribution properties of the molecule. So once you inject the radiolabeled peptides in the bloodstream, through the blood circulation, the peptide itself can specifically find the receptors on the cell surface and then bind there. So you can generate, the peptide can generate signals, radiolabeled peptide can generate signals for SPECT and PET imaging. This slide gives you an idea about how a radiopharmaceutical can essentially find the targets and accumulate in the tumor sites in the body. So basically you need to inject a radiopharmaceutical first into a patient through the vein. Through the blood circulation, the drug will distribute throughout the body of the patient. And as time goes on, the drug clears from the non-tumor tissue rapidly because there's no target available for the radiopharmaceutical to bind. And eventually the radiopharmaceutical remaining in the body is retained in the tumor site because the peptide will find the receptor and bind there. This slide gives you an idea about the clinical imaging process. First of all, you need to inject a radiolabeled imaging probe into a patient, and after a certain period of time, the patient will lie down under the camera for data collection here. And the data collection can be can generate a lot of projections here, and through the reconstruction process, it can generate tomograms here. Now the radiologist will come and look at these pictures and diagnose the disease. So now the question is, can we do this type of imaging in the small animal? The answer is yes. So here I show you the nanospec CT available in our college, which can be used for the small animal imaging. So as you can see, this machine is not too big. It's about 1 meter wide and 1.6 meter tall, and we can use for mice imaging, rat, and rabbit. I think we may be able to do small monkey as well by changing the size of this hole. And here shows you two examples using the imaging probe developed in my laboratory for the primary melanoma imaging as well as the pulmonary metastatic melanoma imaging here. So as you can see, to generate such specific cancer images, you need to have receptor-targeting or cancer-specific imaging probes. We also have a lab PET, small animal imaging PET, we can use for PET imaging. Here gives you an example by using F18FDG into a small mouse to detect the pulmonary metastatic melanoma lesion here. So you can easily, through the CT image, you can see the spot here, and this is a necropsy picture of the lung. And essentially, the F18FDG can detect this spot as a lung mass here. But despite the success of F18FDG, you can see F18FDG can generate a very high background here. The reason for that is F18FDG is not a cancer-specific imaging agent. Okay? The elevated uptake of F18FDG in cancer cells is because cancer cells normally have higher energy consumption compared to normal cells. Okay? So now, let me give you a couple examples about how to targeting the gonadotropin releasing hormone GnRH receptor for breast cancer imaging. First of all, I'd like to give you a brief introduction of breast cancer. The breast cancer is the most commonly diagnosed cancer in females. And the second leading cause of cancer-related death among women in the United States. About 209,000 new cases were diagnosed, and about 40,000 cases of death occurred in the United States in 2010. And normally we use mammography to detect breast cancer. And mammography is a very effective detection tool for the primary breast cancer. However, it cannot detect the metastasis of the breast cancer. And F18FDG, as I briefly mentioned, is also a most commonly used PET imaging agent for the cancer detection, including breast cancer. So basically, F18FDG can be also used for some other cancer detections. And F18, like I mentioned, F18FDG is not a cancer-specific agent. So it has limited applications in detecting breast cancer metastasis because of the lack of specificity. And in this project, we're interested in to use GnRH receptors targets and to develop imaging agents. And because these GnRH receptors are overexpressed on the breast cancer cells, and as well as overexpressed on the human breast cancer samples. And very importantly, the GnRH peptide can bind to the GnRH receptors with nanomolar binding affinity, making it feasible to use peptide to specifically deliver imaging isotope for breast cancer imaging. So as you can see here, this is a native GnRH peptide sequence, and both N-terminals and the C-terminals are involved in the GnRH receptor binding. So if you want to attach a metal chelator to this molecule for radiolabeling, basically you don't want to attach either N-terminus or C-terminus because both are very important for the receptor binding. So our hypothesis was the GnRH peptide can specifically bind to the GnRH receptors on the human breast cancer cells, and we can use it to deliver some diagnostic radionuclide, specifically indium-111, to breast cancer cells for breast cancer imaging here. So as you can see, because both N-terminus and C-terminus are involved in the receptor binding here, therefore we choose to conjugate radiometal chelator here through the D-lysine here, either through a linker. As I mentioned, different linkers can be used either to optimize the binding affinity as well as modify the biodistribution properties of the whole molecule. So first of all, to do the proof principle studies, we designed 3 peptides, try to answer this question. Because the lysine has 2 amino group, alpha amino group and an epsilon group, amino group. So the first question when we tried to synthesize peptide was, which group is better? We don't know that. Therefore, we kind of put the metal chelator through the linker, either on the epsilon amino group of D-lysine or on the alpha amino group of D-lysine. And we also switch the D-lysine to L-lysine to see if the configuration can also affect the binding affinity. So essentially, by evaluating these 3 peptides, we want to answer the question is which amino group is the best in terms of the delta coupling. Here shows you the peptide synthesis and the identification here. Essentially, I'm not going to focus on the chemistry part here. Essentially, we use FMOL chemistry to synthesis a peptide. Here, just give you one example, which is DOTA AHX D-lysine 16RH1. So essentially, we conjugate the metal chelator DOTA to the epsilon amino group of D-lysine through a AHX linker here. And after the peptide synthesis, we use HPLC to purify it, and also use mass spec to identify the peptide. Then the next thing you want to do is, the next question you want to answer is if the conjugation of the DOTA metal chelator really affects the binding affinity of this molecule or not. So essentially we did a competition study here, competitive assay study here. And essentially you can see 3 peptides display different receptor binding affinity, essentially. And the DOTA GnRH1, which was the metal chelator was conjugated to the epsilon amino group of D-lysine through the AHS linker, displayed the highest receptor binding affinity here, which was about 36.1 nanomolar. So essentially through this in vitro study, we can conclude the conjugation of the DOTA to epsilon amino group of D-lysine maintain the nanomolar receptor, GnRH receptor binding affinity. So after that, we just focus on that peptide because it displayed 36.1 nanomolar binding affinity. Here shows you how to prepare indium-labeled compound. So you want to put a radionuclide to the peptide now. For essentially, you can do spectral imaging. The synthesis procedure is pretty straightforward. Basically, you can put the peptide, radionuclide, and in a buffer, and incubate at 75 degrees for 45 minutes. Then through the HPLC purification, we get a very single peak here for the imaging. And just to remind you, the decay properties of the indium-111. Indium-111 is a good spectral imaging isotope. It's being widely used in the clinic, in nuclear medicine department. And it has a 2.83 days half-life. When indium-111 decays, it can generate 2 gamma rays, 171 and 245 keV gamma rays. You can be used for the imaging purpose. And by the way, an indium-111 is commercially available, so you can easily just order from Nordion or Melanoma maybe to do your study. Another important primarily we need to determine its thermal stability. So you want to make sure the peptide you designed is stable enough for them to find the receptor to bind to the receptor. So here shows you the stability of indium-labeled DOTA AHX GNRH1 here. This is a QC after the HPLC purification, and this is 2-hour serum stability data. This is 18-hour serum stability data. So pretty much, as you can see, this peptide was stable in the mouse serum. That's 37 degrees to up to 18 hours. And we also want to show the specificity of this tracer to bind to the GNRH receptor listed here. So as you can see, without a peptide block, with the peptide blockade, the binding to receptors, GNRH receptor can be blocked by 85% here. So if you put a cold peptide to block the receptor sites, then the binding drops down to about 15% of the original one, demonstrating this tracer, even after you label it with indium-111, can still display GNRH receptor specific binding. Now, this slide shows you the GNRH receptor expression on both cells and the xenografts. So we choose MDA-MB 231 human breast cancer cells as cancer cells to generate animal models to further evaluate the biodistribution properties of the radio tracer. So as you can see here, GNRH receptors are overexpressed both on the cells and the tumor xenografts here, making it a suitable model for us to evaluate the tracer we develop. This slide shows you the biodistribution of this indium DOTA GNRH1 here. And as you can see, this tracer displays a very fast tumor uptake here. 0.5 hour post-injection, it reached about 1.8% ID per gram in the tumor. And so demonstrate possible we can use this one for the cancer imaging here. Then we further determine the human breast cancer imaging using the spec CT we have, and use this probe to do the imaging studies. And as you can see here, we did this study with 1 hour post-injection. Basically, you inject the tracer into the mouse through the tail vein, and 1 hour post-injection, you put the mouse on the spec CT to collect all these images. So you can see the backbone of the mouse collected from the CT by CT. So provides you the anatomic information of the whole mouse. And then all this colorful information is from the SPECT. Can provide you the functional information of that tracer. So you can essentially you can see by using this tracer you can clearly see the MDA-MB231 human breast cancer here. And also you can see the urinary clearance here, as well as some kind of liver uptake here. So this is two organs now we're working on trying to decrease down the uptake either in the kidney and the liver. So hopefully we can reach this conclusion. The DOTA position displayed is an important factor on the binding affinity of the GNRH peptide. So essentially the coupling of DOTA to the epsilon amino group is essential to remain the nanomolar binding affinity of the GNRH peptide. And the successful imaging of this human breast cancer xenographic tumor lesions using the imaging probe we developed provide a kind of a platform for us to develop further more imaging probes with improved biodistribution properties here. Now let's switch gear to another type of cancer, which is melanoma. In this type of cancer, we're using different receptors. So basically, I'm going to give you the second example by targeting G protein coupled receptors you can use for the cancer imaging. A brief introduction on melanoma. So melanoma is number 6 commonly diagnosed cancer in the United States. And 1 out of the 75 Americans will eventually develop this disease during their lifetime. And in fact, 1 person dies in an hour in the United States because of the malignant melanoma. And about 68,000 new cases was diagnosed, and about 8,700 cases of death occurred in the United States in 2010. And we're very interested in to develop imaging agent as well as therapeutic agent for this cancer, because there's no curative treatment available for metastatic melanoma. And also melanoma specifically, compared to other type of cancer, is more resistant to the conventional chemotherapy agents. We've been using melanocortin-1, MC1 receptors as targets because this receptor overexpressed on both human and mouse melanoma cells. And as you can see, it's a G protein coupled receptor with 7 transmembrane domains here. And very importantly, the alpha melanocyte stimulating hormone peptide and many of its analogs can bind to these receptors, like shown here, as nanomolar or subnanomolar binding affinities. Therefore, we were very interested in using the alpha MSH peptides to deliver imaging isotope and eventually deliver therapeutic isotope for melanoma treatment as well. Let's look at the Y-type alpha MSH peptide. So Y-type alpha MSH peptide is a peptide with 13 amino acids here, and the amino acids in pink are message sequence for receptor binding. So you have to keep this 4 amino acids there for MC1R binding, as well as in this same order. It's very important. And the NDP-MSH is a gold standard for in vitro binding studies, because this peptide has subnanomolar binding affinity with the MC1 receptors. And several groups have tried label this peptide, linear peptide, with radionuclides for melanoma targeting. And unfortunately, the success was limited because, majorly because of the in vivo stability of linear peptide. So one way to improve the stability, in vivo stability of the peptide is to cyclize the peptide. So these slides give you an idea about different cyclizations have a different effect in terms of tumor uptake and kidney uptake here. And the 3 peptides listed here, the first one is DOTA CCMSH. So you can see this peptide is a linear peptide. And second one is DOTA CMSH. The peptide was cyclized by the disulfide here. And essentially the third one is metacyclized peptide. Now, if you look at the biodistribution data to our post-injection, you can see the linear peptide displayed moderate tumor uptake here, about 5.9% ID per gram at 2 hours post-injection. And the kidney uptake was high, about 63% ID per gram at 2 hours post-injection. Now, when you switch to the disulfide cyclized peptide, which is peptide 2, the tumor uptake was slightly higher, better than the first one, linear one. Very important, if you look at the kidney, you can see the kidney uptake dropped almost 50%, 45% probably. And essentially the rhenium cyclized peptide displayed the best, the highest tumor uptake. Here is about 17.3% ID per gram. And very importantly, you see the kidney uptake further dropped to 8.7%. So this 2 studies demonstrating you can use peptide cyclization to improve the tumor uptake and also affect the normal accumulation of the tracer to the normal organs. And also different cyclization strategy has different effect in terms of the tumor uptake and the renal uptake here. So in my lab, I'm very interested about using lactam bridge to cyclize the peptide. The reason for that is metal cyclization displayed very high tumor uptake, but the synthesis is a little bit complicated. You need to put the metal in it after the peptide synthesis. Alternatively, if you use lactam bridge cyclization, you can finish this cyclization easily during the peptide synthesis. Okay. So specifically, the first generation, we're more focused on designing the peptide like this, more close to the structure of native peptide, alpha MSH peptide. You can see we kept the MC1 binding sequence here, and the peptide was cyclized by a lysine asp lactam bridge here. And the DOTA is a good metal chelator, so we conjugate the DOTA to the peptide either through a linker or without a linker. So when you compare the biodistribution of these two molecules, you can see the effect of the linker on tumor uptake and also normal organ uptakes. And the third one we were interested in if the DOTA position can really affect the binding affinity or tumor uptake or renal uptake as well. So we conjugate the DOTA here instead of this side. And then also you can use this, you can label this peptide with indium 111. Okay, now to evaluate the exact biodistribution properties of the radiotracer, we specifically established two models. This is one of the models, E16F1 melanoma mouse model. Essentially, this model was designed to mimic the primary melanoma development. So what we did was we inject emitting B16F1 cells subcutaneously in the right flank of the mouse. And after 9 to 10 days, the tumor will reach about 0.2 grams here. And then you can use for biodistribution or you can use for imaging studies. Okay, now here shows you the biodistribution of melanoma imaging with this indium DOTA glycolate CYCMSH. As you can see, this peptide has very high tumor uptake. It's very high MC1 binding affinity, about 0.9 nanomolar here. And you can easily see the tumor lesions can be easily visualized here using this tracer as imaging probe by SPECT CT images here. So here is the kidneys and here is the bladder. So you can easily see the urinary excretion pathway of this peptide. So specifically, just to remind you, we have a full biodistribution published in this paper. Just extract the information from that paper. The tumor uptake was about 10.4% ID per gram 2-hour post-injection. And renal uptake was about 13.1% ID per gram at 2-hour post-injection. So this study demonstrates lactam bridge cyclization also works. It works. It can be used to cyclize the peptide for a good melanoma imaging. Oh, so I didn't include the other peptide, which was indium DOTA CYCMSH without linker. So essentially, this peptide with the linker has a little higher tumor uptake compared to that one, and the kidney uptake was less than the peptide without linker. Here shows you the other peptide. We conjugate DOTA at a different position here, and also the binding affinity was high, 0.6 nanomolar, and you can easily see the tumor lesions here as well by using this imaging probe for SPECT CT images here. And specifically, if you look at tumor uptake, it was a little slightly higher but comparable, 11.4% ID per gram at 12 post-injection. And surprisingly, we had a very high kidney uptake here compared to the previous one we talked. It was about 60.5% ID per gram. So essentially, what I'm going to, the taking home message is even you have a very similar binding affinities, you can't really use in vitro data to predict what's the tumor uptake or the normal organ accumulation will be. So biodistribution studies are necessary. So now you may ask me a question, can you label the peptide with some other imaging isotope? The answer is yes. So this gives you an example. Basically, we can switch indium-111 to gallium-67, which is also a SPECT imaging isotope here. So also by switching indium-111 to gallium-67, this peptide also displayed good tumor uptake here, about 12.9% ID per gram at 12 post-injection. You can easily see tumor lesions clearly here. But compared to the indium compound we just talked about, the kidney uptake was a little higher, so it's about 27.6 ID per gram at 2-hour post-injection. And another potential application for this study is you can easily switch this gallium-67 to gallium-68, which is a generator-produced PET isotope for the PET imaging. Okay, I mentioned we established two models. So another model is B16F10 metastatic melanoma model. Try to mimic the metastasis development in human. What we did is we inject this melanoma cells into a mouse through the tailbone. Try to mimic the cancer cells detached from the primary site, now get into the bloodstream, through the blood circulation, try to establish metastasis in some other organs. So essentially, because we inject into the tailbone, the blood will pump back to the heart, then pump it out. And leave, actually the lung will be the first organ to develop the metastasis. But as time goes on, if time is long enough, you can still see metastasis either in the bone, liver, adrenal gland, and some other organs. So here shows you the left panel shows you the CT image of a normal mouse. And after the CT image, we took out the lung here. So here shows you an biopsy picture of a normal lung here. And the right side shows you a CT image of a mouse bearing lung metastasis here. Okay? And this is the lung we took out after the CT image. So if you look at the CT image, you can see for the normal mouse, the lung was functional, 100% functional. You see a lot of air in the lung area, dark area here. But, you know, for this mouse bearing the lung metastasis, the lung function was not 100%. So you see some of the lungs still working, but most of them didn't work. You don't see a lot of air in the lung area. So in other words, you can use micro CT to monitor the development of the metastasis. Okay, now, so to answer the question if we can use our imaging probe to detect metastasis, we did a study with our imaging probe. And meanwhile, we try to answer the question how good it is compared to F18FDG. So essentially, we inject a melanoma-bearing mouse with the F18FDG first day, we inject it in, and then we collect the PET imaging of this mouse. After study, we took the mouse and put it back in the cage, waiting for another day. So the reason for that is we try to wait the F18 decay. So half-life for F18 is 2 hours. So after a day, it decayed away. And then we took the same mouse back and injected with the indium probe we developed here, and use the SPECT imaging to collect the SPECT imaging, and then compare to see how easy or hard we can find the metastasis. So as you can see the accumulation of the activity in the lung area by the F18FDG image, but you cannot easily see the individual spots developed here. On the other hand, the SPECT imaging using the indium probe we developed, you can easily see all the spots developing in the lung, the melanoma metastasis here. So indicating in the small animal model and our receptor-targeting imaging probe is better than the F18FDG to pick up all this small metastasis developed in the lung. And another study we also conducted was we tried to compare two animals bearing metastatic melanoma to see if we can use it to monitor the development. So essentially, this mouse was 20 days post the cell injection here. As you can see, as time goes on, you have more metastasis develop here. Also, the lesion kind of became bigger here. In other words, what we try to demonstrate is if you start a chemotherapy or some other therapy treatment here, what did I do? Sorry about that. So if you start a treatment, then you can use this imaging probe to monitor the therapeutic response to see the shrinkage of the lesions. And then we further did a study to try to answer how early we can use this imaging probe to detect metastasis. So I've showed you the 17 days development and 20 days development. This study we did was 13 days post IV injection. So you can see there were only 5 individual spots in the lung By using the imaging probe, we can also clearly detect this metastasis here. And this is F18 PET imaging after 16 days post-development. Okay, let me talk about the second generation of the lactam-bridge cyclized peptide for melanoma imaging. So if you still remember the biodistribution I showed in the early slides, one question we have after we kind of developed the first generation peptide was the tumor uptake for this peptide was not as high as the metacyclized peptide. So the metacyclized peptide displayed 17.3% ID per gram 12 post-injection, and the tumor uptake for the first generation peptide is about 10.4%. So one question was, we have demonstrated lactam-bridge cyclization work. You can still develop this type of peptide for melanoma targeting, but the question was, can it be as good as metacyclized, or can it exceed the metacyclized peptide? So we just simply looked at the structure and said, okay, the direct difference is, the cyclization, the cyclic moiety is different. So for this one, we have 12 amino acid in this moiety, and the metacyclized we only have 8. So why don't we just try a smaller one? We don't know if the size is an important factor at that time. And what we can do is just cyclize, we just kept the MC1 binding sequence here, but how about just cyclize it directly using this ASP less lactam-bridge? So that's what we did. But we were not sure if this is good or not. But after the in vitro binding assay, we said, okay, it's good. We're worried about this kind of a cyclization may affect the binding region very badly. So in terms, after we got the binding affinity here, oh, it's still good. 1.8 nanomolar binding affinity is still kind of a very high binding affinity. So this is the peptide I just talked about. And meanwhile, we try to use different type of linkers to answer two questions. The first is, we don't know if the NLE cyclic moiety is very important or not. Can we insert amino acid in it to modify the biodistribution properties, or we have to insert amino acid between DOTA and the NLE instead of this between NLE and the cyclic moiety? So we synthesized two peptides and determined IC50. It was interesting to see, actually, it's not a good idea to insert the amino acid linkers here to separate NLE and the cyclic moiety. Because if you did that, the binding affinity dropped to 873 nanomolar. But if you move the amino acid linker more towards the DOTA, then the binding affinity comes back at 11.5 nanomolar. Okay? And also we did this peptide. If you change this glue amino acid to glycine, the binding affinity comes back around 2.1 nanomolar, very close to this peptide without any link. Therefore, we decided, okay, let's do the biodistribution for these two peptides to see. So here shows you the biodistribution and the imaging of this indium DOTA NLE CYC-MSH hex. So basically, we don't have any more amino acid linker here. After we saw the biodistribution, we were thrilled because if you look at the tumor uptake, now it's 19.4% ID per gram 12 post-injection, even slightly higher than the metacyclized peptide. So it seems like the smaller ring kind of works better compared to the bigger ring in terms of the tumor targeting. And importantly, if you look at the kidney uptake, now dropped to 9.5%. So in other words, it increased the tumor uptake, also decreased down the renal uptake. And now you can see for the image, the contrast is better than the first generation peptide here, and you can easily see the lesion, flank lesion clearly here. Okay. If you look at the tumor to kidney uptake ratio, compared with the metacyclized peptide in the first generation, you can better understand the improvement by switching to the second generation peptide here. So the blue columns are indium DOTA glycosyl message, which was first generation. And this green columns are metacyclized peptide. Yellow columns are second generation peptide without linker. So essentially you can see by switching to this second generation peptide, it displayed comparable high tumor to kidney uptake ratio compared to the metacyclized, very comparable with metacyclized peptide. So here shows you the biodistribution of the other peptide with the GG linker with the similar binding affinity, 2.1 nanomolar. And you can see the tumor uptake was high as well, so about 18.5% ID per gram at 2 hours post-injection. Very importantly, all this normal organ accumulation were lower than 1.5%, I would say. And importantly, you see the kidney uptake now is about 7.5%, so even further lower than the peptide without the glycolide linker. And you can easily see the melanoma lesions clearly with all these individual spots here. A lot of more information. So this slide shows you the comparison in kidney and liver uptake between the peptide with GG linker or without GG linker. So basically the blue curves are Indian peptide with the GG linker in it. So quickly you can see by insert this GG linker, it decreased down the renal uptake about 1/3, 30%, and the liver uptake was decreased about 2/3, 60%. Now, if you look at the tumor to kidney ratio, tumor to liver uptake ratio, it's another way to really see the improvement. So by insert a glycolide linker, you kind of increase the tumor to kidney uptake ratio as well as tumor to liver uptake ratio here. So this is very important because one of the directions we want to work with this peptide is try to use the peptide to do the treatment. So you can easily switch diagnostic radionuclide to therapeutic radionuclide. For the therapeutic perspective, then the ratio is important. You want to deliver the maximum dose to the tumor and the minimum dose to the normal organs, right? So the improvement in tumor to kidney ratio, tumor to liver ratio were significant. So in conclusion, so we have developed 2 generations of the MC1 receptor-targeting peptides. Both primary and metastatic lesions could be visualized by the first generation peptide. I didn't show you the peptide distribution of the peptide without linker. But basically, increasing the overall negative charge of the peptide could significantly reduce the renal uptake of the peptide without affecting the tumor uptake. And we found the reduction of the peptide size essentially contribute to the tumor uptake, increase the melanoma uptake of the second generation peptide, also decrease down the renal uptake. Amino acid linkers exhibited profound effects. So if you look at the peptide with the glycolide linker or without glycolide linker, you can easily see by insert the amino acid linker, you can further decrease down the renal uptake as well as the liver uptakes. So I'd like to thank postdocs and graduate students working in my lab and the collaborators in our cancer center and the financial support to this project. I'd like to thank you for your attention. I'd be happy to answer any questions. Yes?
You mentioned that you could just take an image and then have things decay and then keep injecting them with image over days. Is there no negative side effect of having these radiolabeled?
So you're talking about the study we did with the F18FDG and the Indian labeled peptides. So one thing I'd like to get your attention is F18FDG, the targeting mechanism is different with the peptide we injected in. So F18FDG is not targeting the MC1 receptor. Okay. So basically, the elevated uptake is because cancer cells has higher antigen absorption compared to the normal cells. So they're targeting different things. And And the peptide we developed there is targeting the MC1R. So I assume there's no cross effects between these two agents. So another thing is, if you want to do the dynamic image over days, you can select the imaging nuclide, diagnostic nuclide with longer half-life. So you can, for example, if even this Indian peptide, you should be able to do it. Half-life is about 3 days. So I'm sure you can do 24 hours post-injection. Same mouse, if you inject this probe, did a 1 hour 12 image, and then you can put it back, and then come maybe 24 hours later to do another image. The only downside is if the peptide is too quickly clear out of the body, then you will lose activity. Okay? Or you can inject a little more to make it last longer. Yes.
We're focusing a lot on minimizing the kidney uptake so that the signal will be lower there. It's never gotten to the point where is the signal low enough that if any of the tumor metastasizes around the kidney or near the kidney, can you distinguish tumor just from kidney signal?
That's a good question. Actually, in this case, because the peptide size, so the excretion pathway is the urinary system to the kidney. The kidney uptake was not receptor mediated. So we have done a blocking study to show you can block the tumor uptake, but you cannot block the renal uptake. So it's non-specific. So in terms if you have some metastasis in the kidney, in this case, I don't think we can use this agent to differentiate that because the high, very kind of a high uptake in the kidney. The reason we want to reduce down the kidney uptake is more focused on the therapeutic perspective. So in this case, if we switch to the therapeutic radionuclide, we want to deliver maximum dose to the tumor or less dose to the kidneys to avoid toxicity to kidneys. Okay.
So just to follow up, the kidney uptake you say is not specific, but it contains in the kidney longer than the tumor. So is the chelate irreversible down? It becomes irreversible down, or what is that?
There are several factors affect the kidney. First of all, it's not MC1 receptor mediated, and the charge has been a major factor contribute to the kidney uptake. So for example, in this case, if we inject some positive charged amino acid, like lysine or arginine, you can block the renal uptake up to 75% in all cases. And also, recently, some of the paper published talk about some of the also receptor kind of involved with a broad spectrum. So we didn't know exactly how this MC1, this peptide kind of interacts.
So you think it may be some receptor-mediated process, but not
the MC1R receptor mediated process, yes. Charge is a major factor. So normally we use charge to, we use positive charge amino acid to block. You can successfully block up to 70% of the uptake.
So when you've got lower kidney uptake, you want you to see regional differences in the kidney activity. Does that correspond to the structure?
So actually, most of the activity in the kidney are on the cortex area, not inside. So I think for that it's just maybe charge interactions. So the positive charge for the peptide side can interact with the negative charge of the kidney cells on that area. Yes?
The structure of your breached or non-breached, is it affect the stability of enucleate?
No.
No?
No.
They're very stable.
Yeah, so what happens is for the linear peptide, like an NDP peptide, you can label with the DOTA. You put the DOTA there and label with Indian 0.11. And what happens is enzyme can kind of find the amino acid and chop it up. So the peptide itself don't have enough time to deliver.
So that's why you have the D types.
Yes, you can. Another way I didn't mention, cyclization is one way, but you can switch from natural amino acid to D amino acid. Can also help increase in vivo instability, because when you switch from L configuration to D configuration, enzyme has hard time to really recognize that and chop the peptide. Yes?
Yes, I would like to go back to the study where you compared the PET and SPECT image of the pulmonary metastasis. And you suggested that the SPECT, those speckles on the SPECT, they are the metastatic cells, right? So did you try to do the pulmonary radiography just to make sure that it's really what you're observing? Because from my perspective, from my experience, the SPECT image very often when you're doing reconstruction produces those speckles, which are nothing real. They are just some artifacts.
We did that studies. So we use autoradiography as well as we take the lung out, put on a piece of paper, lie out, and did the SPECT imaging for that as well. So we include that image in the paper published. Identify all these spots you can easily see. We did that as well. Autoradiography and the SPECT as well.
And the second question which I had is the first study would be nanotropin receptor. So you showed that the SPECT image, but also those receptors are expressed in breast and lymphocytes on the prostate and also on the ovaries. And I didn't see those actually organs light up. So could you comment on that?
You're right. So this GRH receptor not only just overexpressed in breast cancer cells, also prostate cancer. The mice we used, first of all, we used the female mice. Okay, so
the ovaries.
Yes, you're right. But from image, we didn't see the uptake there. But unfortunately, we didn't really take out the ovary to count. So from biodistribution, we should be able to see that. But we didn't do that study at that time.
Maybe it's the question of the contrast. If you increase the contrast of the image, maybe it will show.
Might be.
Is it known what the expression levels are?
Yeah, maybe the yes, John, that's a good point. The expression level may be different as well. But we haven't really focused on ovarian cancer yet. But it's been reported. I think one of the cell lines people has used for is OVA3 we can use for this GRH receptor. They reported the expression, but the level I don't have the detailed data about compared to the density. Is that cancer cells or ovarian cancer cells? Yeah.
When you switch from indium to gallium, once you decrease the kidney uptake? Increase. So it's not issue in the chelate step because indium and gallium.
No, normally when we switch isotopes, sometimes you don't see any difference in terms of renal uptake or kidney uptake. But sometimes you do see either in the tumor uptake or the kidney uptake. We don't know exactly the reason. The coordination chemistry is a little different though, may contribute to this. So
because in copper chemistry, the copper chelation of DOTA or NOTA, that stability is critical in the based on pH and the serum stability. But is there any study for indium and gallium?
Not for this one. So DOTA is a very good chelator for indium and gallium. Very good chelator. In terms of the copper-64 mentioned, specifically DOTA is not an optimal chelator for copper-64. The reason for that is once it's get in the bloodstream, and some of the enzymes will compete of the copper-64. So that's why I don't know if been to my first talk, we talked about the copper-64 with DOTA and with NOTA. And you can see some of the very high normal organ accumulations there. That's because some of the enzymes coordinate with the copper-64. But when you switch to NOTA, it's very stable. That case is very stable. And then easily through the biodistribution comparison, you can see there's no, the normal organ accumulation are very, very low, less than 1% microgram in our case. But for the indium and the gallium, then DOTA has been a very good chelator for both within it. Any more questions?